Okay, so we've discussed two mechanisms by which you can get drug resistance to antibiotics. Now let's discuss a third. So another way is that you can enhance DNA repair. Now you might be wondering, well, what use is that? But what you need to realize is that some antibiotics that we use to attack bacteria, admittedly, they're not nice antibiotics, they're horrible antibiotics, but some antibiotics that we use to kill bacteria target DNA and they damage DNA, and by damaging the DNA of the cell, they kill the cell, basically. Okay, so a way in which bacteria can become resistant to such drugs is by enhancing the mechanisms that they have for repairing DNA, i.e. by increasing the expression of the DNA repair machinery to repair the damage that these drugs are doing. Okay, so there's a mechanism by which you can resist DNA damaging drugs. Okay, fourth mechanism is something known as metabolic bypass, and I suppose this is kind of similar to enhancing DNA repair. So metabolic bypass, let me give you an example of metabolic bypass. Well, firstly, actually, I'll give you an example in terms of peptidoglycan transpeptidase, this one that we've seen, and then I'll give you a really archetypal example. So metabolic bypass basically means the bacterium says, hey, look, you're throwing this drug at me. This drug is taking out my enzyme. So, for instance, let's say we're throwing penicillin. Penicillin is inhibiting the peptidoglycan transpeptidase. So, what does the cell do? Peptidoglycan transpeptidase is no longer functioning. Does it just lay down and die? No, it doesn't. Instead, it produces more peptidoglycan transpeptidase, so it increases the expression of peptidoglycan transpeptidase. So it's going to be frantically trying to produce more of this enzyme and sending it to the cell wall. Okay, if that did happen, if it could produce enough peptidoglycan transpeptidase that actually it could cope with the fact that the drug was inhibiting its peptidoglycan transpeptidase, i.e. the rate at which the drug was taking out peptidoglycan transpeptidases was the same rate at which the bacterium was making new peptidoglycan transpeptidase. That would be an example of metabolic bypass, where effectively the drug's working, but the bacterium is just producing so much more of the target that the fact that the drug is taking out some of the target just doesn't make any difference. It's producing so much that it's still got enough to be functional, but to have a functional output from that population of enzymes, basically. Okay, so now I'm going to give you another example of this, a very famous example. So there is an antibiotic known as trimethoprim, which is a synthetic antibiotic. And basically, trimethoprim inhibits the bacterial dihydrofolate reductase enzyme. Okay, and I want to stress it inhibits the bacterial dihydrofolate reductase enzyme. It does not inhibit the human one, which is very, very convenient because this enzyme is very important. So dihydrofolate reductase catalyzes a reaction, basically, and the reaction it catalyzes is the conversion of dihydrofolate into tetrahydrofolate. Okay, so this dihydrofolate reductase enzyme converts dihydrofolate into tetrahydrofolate. Tetrahydrofolate is important for the synthesis of thymine nucleotides, basically. So if you want to make DNA, you need to have the nucleotides that you're going to use to make the DNA. And you're going to need 4 different types of nucleotide. An adenine nucleotide, a thymine nucleotide, a guanine nucleotide, and a cytosine nucleotide. Basically, to create the thymine nucleotide, to create deoxythymidine monophosphate, which is the nucleotide which you're going to use to make DNA. you need tetrahydrofolate. If you haven't got tetrahydrofolate, you can't make this nucleotide. You can't make the thymine nucleotide, basically. Okay, so if you don't make the thymine nucleotide, then you can't copy your DNA. If you haven't got enough nucleotides, then how can you copy the DNA? So basically, if you give this drug to cells which are dividing very, very rapidly, such as bacterial cells. So these are bacterial cells dividing very, very rapidly. Every time they divide, they have to completely copy their DNA, and then they'll divide, basically. If they cannot synthesize the nucleotide for thymine, this deoxythymidine monophosphate, then they're not going to divide, because if they can't synthesize that, they can't copy the DNA. And if they can't copy the DNA, they can't divide. So this drug is very effective at blocking bacterial cell proliferation, basically. So it stops these bacteria from being able to divide. Okay, but a way in which the bacteria can get around trimethoprim inhibiting this dihydrofolate reductase enzyme is to just increase the expression of dihydrofolate reductase. And by the way, dihydrofolate reductase is often abbreviated to DHFR, and of course dihydrofolate is often abbreviated to DHF, and then tetrahydrofolate is often abbreviated to THF. Okay, so what it can do is it can just upregulate this dihydrofolate reductase. And if it makes so much of the enzyme that even though you've inhibited loads of it through this trimethoprim, you still have enough to synthesize enough tetrahydrofolate that you can make enough deoxythymidine monophosphate in order to actually proliferate, then that's a way of resisting this drug. You haven't changed the target, you haven't destroyed the drug, you've just produced so much of the target that even the amount of drug you're receiving is not enough to inhibit it all, basically, and you still get a certain amount of functional activity by that target, enough to produce enough of the product, basically. Okay, so that's metabolic bypass. And then finally, another mechanism is that you can have drug efflux pumps, okay? Or you can reduce the drug entry to the cell. Okay, so drug efflux pumps is an excellent example. So basically, what cells can do, what bacterial cells can do, is they can start expressing pumps in their membrane. So let's say this is a pump here, which is going to take the drug from the cytoplasm and chuck it back out of the cell membrane, basically, well, back out of the cell. So you can start getting the expression of these drug efflux pumps, i.e. when you put the bacteria in this medium containing the drug, what they will start doing is making these drug efflux pumps, putting them into their cell membranes, and the drug efflux pumps will be extruding the drug actively from the cytoplasm and basically taking the drug down in the cytoplasm. Okay? Alternatively, you can employ mechanisms that just mean that the drug can never get in. So potentially the drug has a specific route by which it gets in to the cell membrane, which may well use some protein in the embedded in the cell membrane. And what the bacteria could do is it could modify that protein so that the drug is no longer able to use that route into the cell cytoplasm, basically. So either ways of extruding the drug from the cytoplasm, such as drug efflux pumps, or ways of making sure it never comes in, so reducing drug intake. Those are both final ways in which you can become resistant to antibiotics, and they are seen, all 5 of these examples are seen in response to antibiotics.